right? Because it's all about the microenvironment. So here's the cell, right? We end up with lots of protons because we're in an acidic environment. So what the cell will do is he's like, listen, I can't change everything, but I can try to change my immediate environment. So he will actually start to produce proteins and move them to the surface of the cell that all have an acidic, like, what do you call that? End group. Which sounds sort of weird, right? But what is that acid gonna attract? A negative charge, right? A positive charge is gonna attract a negative charge, which means all the hydroxyls from solution are gonna come and they're gonna buffer this little portion of the environment, which is pretty slick. He knows he can't change the pH of the whole ocean, so he doesn't try. He just tries to change the pH of just that little bit of space around him, right? So they will either, right, they will gather protons to offset an acidic environment, or they can gather hydroxyls, or sorry, they will gather hydroxyls by having positively charged proteins in an acidic environment, and they'll do the opposite in an alkaline environment, right? They also, like you, have a buffer system. Your buffering system is CO2, right? You can make your CO2 move from CO2 to carbonate back and forth and back and forth and back and forth, which is why you'll hyperventilate if your blood pH changes. They actually use their DNA as a buffering system, right? Because phosphate groups are great buffers. So you have a carbonate buffering system, they have a phosphate buffering system. So if the protons get into the cell, they just bind them to the backbone of their DNA. If they don't have enough, they let them go into there. So they buffer their internal environment. Now, what about alkaline environments particularly, right? They actually do, most organisms who live in an alkaline environment also live in a high salt environment. So they will actually use the salt concentration to modify the pH. They will use it to pump. You know, in general biology, you learn about active transport, and you learn about the sodium-potassium pump, where one goes with its gradient to pump the other against its gradient. We call that an antiport, because they're going in opposite directions. They actually use the salt in the environment to pump protons to offset that alkaline environment. And they have more adapted surface proteins. In acidic environments, like our Thiobacillus ferrooxidans, who lives in a pH of 2, what do? Right? There you have to have a membrane that's really impermeable to protons so that they can't get inside the cell and digest everything inside there. So they are very impermeable to protons, whereas these guys actually have to pump protons. And then these guys also have surface proteins that adapt. So that's another consideration that we have to do, right? All of the media that we make have buffers in it so that we can keep that pH constant. And for all of the bacteria that we grow, we like to keep that pH constant at about 7 because all of the bacteria that we're growing are typically neutrophilic. And we're going to do a lab where we're going to inoculate bacteria into a low pH, a neutral pH, and a high pH, and we're going to look to see what do they like best. We're going to let them grow and see who grows fastest. And we're going to test that by using a spectrophotometer. We're going to use a spectrophotometer, put each of those tubes into the spectrophotometer, and look at which one's most turbid. The one that's most turbid grew the best, which means that must be its optimum. So we're going to try to determine the pH requirement of a couple of different organisms. And then finally, our third that we're going to test is oxygen, which seems weird, but we actually have to talk about the fact that oxygen is toxic. We breathe it, so we think it's normal, right? Like, oh, it's just oxygen. I mean, I'm breathing it, it's in the atmosphere. Like, what could be wrong with oxygen? Well, oxygen, O2, is a fairly chemically reactive molecule because it really likes electrons. And because it really likes electrons, it will kind of wreak havoc by stealing electrons. And once it becomes negative, then it becomes really, really reactive, right? So whenever you are living in an aerobic environment, you are constantly producing these things that look like molecular oxygen but are more reactive because they're negative, and we call those superoxides. And they're really reactive and really damaging, And sometimes you'll hear them called reactive oxygen species. You'll also hear them called free radicals. And they're radical, man. They run around and they just bang into everything like a Tasmanian devil. They crash into DNA, they crash into RNA, they crash into fatty acids, they crash into amino acids, they crash into enzymes, and they destroy them. And that happens to every cell that is exposed to oxygen all the time, including yours, and including mine, and including E. coli, and everybody else that you've ever grown in there, because everyone you've ever grown, you grew in the presence of oxygen. So if you live in the presence of oxygen, you have to have a way of dealing with this, right? So we have two big ones. Superoxide dismutase is an enzyme, and catalase is an enzyme. And those two work together to keep you safe, you being a bacterial cell, when you live in the presence of oxygen. So when oxygen becomes the superoxide anion, superoxide dismutase will take it and turn it to hydrogen peroxide. And you're like, whoa, 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 superoxide dismutase. Like, come on, man, like that's just as bad. Right? That's not good. You use that to kill bugs then when they get on your cuts, right? So like, great, man, thanks a lot. Now you've taken a problem and you've actually made it worse. But thankfully, the organism who has superoxide dismutase will also have catalase, which, by the way, are both antioxidants. When you eat an antioxidant, that's what you're trying to fight the effects of, is reactive oxygen species in your body. The damage that is done by these reactive oxygen species, which ages your cells and gives you wrinkles and can cause cancer and all sorts of fun things, right? So if you take an antioxidant, that's what you're doing. You're trying to take a crazy, reactive, nasty oxygen species and convert it to something that's not crazy nasty. So superoxide dismutase can deal with the superoxide anion, which is great, but it doesn't take care of the problem completely. It has to work in conjunction with catalase, because catalase will take the hydrogen peroxide and convert it to water. And that's inert. And that is what you have. You have catalase. So when you put hydrogen peroxide on your cuts, the reason they bubble, it's not because the bacteria are like, ah, and they're all dying. It's because your body's like, ah, it's hydrogen peroxide, send out the catalase and get rid of it. So the bubbling you see is because you have catalase in your bloodstream, and it's taking hydrogen peroxide and it's converting to water and oxygen gas. And the bubbles you see are the oxygen, just the molecular oxygen that's given off. So now you can recycle your molecular oxygen and send it to the end of your electron transport chain to collect electrons and help you to make ATP, right? We don't test directly for superoxide dismutase in class, but we do test for catalase because it's super easy. All you have to do is squirt hydrogen peroxide onto bacteria, and if it bubbles, guess what enzyme it produces? If it doesn't, guess what enzyme it doesn't produce? And if it has the enzyme, it has the gene, and now we know something about the genome. For instance, when you are trying to differentiate a skin infection, two major culprits for skin infection are Streptococcus and Staphylococcus. As their name suggests, both are cocci. They are also both gram-positive. So under a microscope, you can't differentiate them because they're both gram-positive cocci. But you wonder, right, they're very different antibiotics that can be used for staph and strep. So you do a catalase test. Staphylococcus have catalase and they will bubble. Streptococcus do not have catalase and they will not bubble. So oftentimes in literature you'll hear catalase negative gram-positive cocci, and they're talking about strep. Catalase positive gram-positive cocci are talking about staph when we're differentiating skin infections. So it's an important clinical tool, and it's one that we're going to be using in lab. We're going to be isolating bacteria from your throat, looking for strep. And one of the ways that we will confirm whether they are likely strep is whether or not they produce catalase. If they produce catalase, they're not likely strep, because strep don't produce catalase. So what that means, right, if you think, well, now strep, they don't produce catalase, and they actually don't really like living in the presence of oxygen. They like it without oxygen, but they can tolerate being in presence of oxygen. Staph actually like it, because they can handle it. Because remember, everybody who can wants to live in the presence of oxygen, because that's going to get the most ATP per molecule of glucose. but it's all a balancing game. You love it, but if it kills you, it's not a great idea. Right? Moderation. So, oxygen, that's our last of the physical growth requirements. We have a super cool medium, right, called fluid thioglycolate medium, which we use to determine the oxygen requirement of bacterial cells. It is the second differential medium that you will be using in this class. The first differential medium you have already used, and that's motility medium. When you inoculate motility media, you can tell whether an organism is motile or not. They'll all be red, but the one that is red and moves away from where you put him is motile. The one that is red and stays where you put him is non-motile. So you can differentiate bacteria based on motility using motility media. You can differentiate bacteria based on their oxygen requirement using fluid thioglycolate media, and you will be doing this first in Lab 11, and then you will do it multiple more times throughout your tenure as a microbiology lab student, because understanding their oxygen requirement tells us something about their genes, and when we learn about their genes, we can identify them, right? So the reason I say this is a really cool medium is because, and we're gonna get a little more in depth here, not really for lecture, but for lab. It has, and this will be good for lecture because we're gonna use some terms that you wanna know for lecture. It has two things in it. It has a reducing agent. Now, knowing what you know about reduction and oxidation, what is oxidation? Losing electrons. What's reducing? Gaining electrons. And how do biologists follow electrons? We look for H's, right? So, oxygen looks like this, right? When oxygen receives electrons, say in aerobic respiration, what does oxygen become, right? It collects electrons in the form of hydrogens and it becomes water. And we say that it has been reduced. So a reducing agent is something that converts oxygen to water, basically making that environment anaerobic. No oxygen is there. The problem is, in a test tube, you cannot see oxygen, right? You can't see it. I mean, if you could, who knows? Because we've been looking through it our whole lives, so it doesn't look like anything to us, right? So in this tube, there may very well be no oxygen, but we can't tell. So in addition to a reducing agent, we need something called a redox indicator. And a redox indicator tells us where we are. Are we at the reduction part or the oxidized part? Are we reduced or are we oxidized? This is the oxidized side. This is the reduced side. So in this medium, we have a redox indicator, which is red when this molecule is in its oxidized form and is clear when it is in its reduced form. So this medium will actually be red on the top because there's oxygen there. And that allows you to visualize where the oxygen is. So then you take your inoculating loop and you very carefully go down and back up, kind of like you did with your inoculating needle in the motility media. The reason you don't shake it is because if you shake it, you'll stir it up and the oxygen will go everywhere, and you have this very nice gradient set up where there's oxygen here and no oxygen here. So when you put them there, there's a bunch of agar, not enough to make it solid, but enough to give them somewhere to sit. And bacteria are like people, we're lazy, we want to sit down, right? They want to find a little home, and so they land on that little piece of agar and they start to grow. And they grow and they grow and they grow and it gets turbid. And now you have growth either where there is no oxygen or where there is oxygen, and that tube can tell you whether or not your organism likes oxygen and requires oxygen or hates it and is killed by it, right? So if you have an organism who only grows at the top where there is oxygen. He's an aerobe. In fact, he's a strict aerobe or an obligate aerobe. He is obliged. He strictly only lives where there is oxygen. Right? Don't worry about the rest of it. Let's go over to this one. Here we have an organism who grows only where there is no oxygen. So we call him an anaerobe, a strict or obligate anaerobe. He cannot grow in the presence of oxygen. Why? No catalase, no superoxide dismutase. Why can he? Because he has catalase and superoxide dismutase. So those are our two extremes. We have aerobes and we have anaerobes. But then notice we have these other, we have this one here, and this one here. So let's take these two. These two can grow where there is oxygen, and they can grow without oxygen. So because they can grow where there's no oxygen, we will call them an anaerobe. But we have to make sure that in that name we also let you know that they can grow with oxygen. So they're not just anaerobes, they have some qualifier. So this guy right here grows where there's no oxygen, but he grows better where there is oxygen. So we call him a facultative anaerobe. So facultative and troph and tolerant all give us some idea, right? So halo tolerant, cyprotroph, facultative anaerobe all say to us, I can, but I don't prefer it. And it's just a bummer that 3 different people gave them names, they all gave them different names, right? But they all mean the same thing. Halo tolerant, I can, but I don't prefer salt. Cyprotroph, I can, but I don't prefer cold. Facultative anaerobe, I can, but I prefer oxygen. And the way that you can tell that is because he grew faster. It's more turbid there. So a facultative anaerobe can grow anaerobically, but does not prefer to grow anaerobically. Now, if we go back to chapter 5, what we would say is he undergoes aerobic respiration here. He gets 38 ATP per molecule of glucose, which allows him to make DNA and make RNA and make proteins and make ribosomes and make cell wall and undergo binary fission really fast, optimally. Here, he's either undergoing anaerobic respiration or fermentation. He's not getting as much ATP per molecule of glucose, so he's growing more slowly because it takes more time to build up all of the things that he needs to undergo binary fission. This guy grows evenly throughout. So he's an anaerobe who tolerates oxygen. He never uses it. So even in the presence of oxygen, he will not park oxygen at the end of his electron transport chain. He will always either ferment or he will undergo anaerobic respiration. He will never use oxygen. And so we say he tolerates air. He tolerates oxygen. He doesn't use it. So an aerotolerant anaerobe grows in the presence of oxygen, never uses it. And then this one over here is kind of a wonky little guy. He's like Goldilocks. I don't want there to be no oxygen, but I don't want there to be too much oxygen. I like just a little bit of oxygen. And we call that micro-oxygen. Micro-aerophiles like a little bit of oxygen. They like it, so if atmospheric oxygen is 20%, they like it like 5 to 15%. And we can determine all of this from the inoculation of that organism into that broth. We inoculate him, go down, go up, put him in the incubator. Don't need any special equipment, no special incubators. Just allow him to grow and determine what his oxygen requirement is. And we will do that in Lab 11 also. So in Lab 11, we will not test salt, but we will test pH, oxygen, and temperature optima. We will try to decide, what do you like best? Then we get to have some fun and try to kill him. Now we know what you like, now we're gonna take you away, right? If we know you like oxygen, what's one way that we can keep you from spoiling our food? Take away the oxygen, right? That's what we do in canning, right? When you can have like a thing of pickles, right? Or you have a stainless steel can of like soup, there's no oxygen in that. Because they know that the food spoilage organisms, right, if there were oxygen, they would like that and they would grow, so they take away the oxygen. Now, the other thing is much easier. Chemical growth requirements, same thing as you. You ready for this? Carbon, nitrogen, sulfur, phosphorus, that's what you like, and vitamins and minerals. Just like you take a multivitamin, trace elements are minerals, organic growth factors are vitamins. So they need the same things you do. They need carbon because carbon is the backbone of every organic molecule. Whether I'm trying to make a fat or a protein or nucleic acid or a carbohydrate, I need carbon. If I want to make a nucleic acid or I want to make a protein, I need nitrogen. If I want to make proteins, I need sulfur. If I want to make nucleic acids, including ATP by the way, I need phosphorus because the DNA molecule is a sugar phosphate backbone. And I need vitamins and minerals because those things, right, they help my enzymes to work the way that they're supposed to work, right? And proteins, of course, are used all over the cell. The flagellum is made of protein, the cell wall has protein, there are enzymes and proteins in the cell membrane, there are enzymes all over the cell, the ribosomes are partially composed of protein, so we need, right, if we need carbon, nitrogen, and sulfur to make proteins, without carbon, nitrogen, and sulfur, we don't have any of these things, right? We also need DNA and RNA. So to get those, we need nitrogen and carbon and phosphorus. And if we don't have those, no DNA, no RNA, no ribosomes, because ribosomes are a protein RNA complex. We need polysaccharides, which means we need carbon. And without polysaccharides, no cell wall, no lipopolysaccharide outer membrane, no storage granule, which means you can't fuel metabolism, which means you die. If we want to make lipids, we need carbons. If we want to make phospholipids, we need phosphorus. And without lipids, we don't have a cell membrane, we don't have a lipopolysaccharide outer membrane, and we don't have a long-term storage fuel that we can use to fuel ATP production. So we will examine a lot of different physical growth requirements, right? Oxygen, temperature, pH. We're not going to get into the crazy of the chemical stuff, so I'm just going to show you how important minute changes in feeding change drastically the outcome of experiments. So this, see some more of my data. You're the only people that ever have seen it. Congratulations, right? This is growth over time of an organism. I'm trying to figure out how to grow him quickly. So I give him a source of nitrogen. Why? So that he can make DNA and RNA and amino acids. I give him this particular nitrogen compound, and his generation time is 85 hours. Yours is 20 minutes, mine is 85 hours. You know how long it takes to get out of graduate school when you have a bug that grows and replicates every 85 hours? Like, you guys inoculate and come back tomorrow, I would inoculate and come back in 2 weeks. So I want to figure out a way to make it faster, right? So I start tinkering with things. One of the things I tinker with is his source of nitrogen. I'm like, you know what? Maybe he doesn't like ammonium chloride. I'll give him glutamine instead, right? That's an amino acid. Maybe that's a little easier. And lo and behold, his generation time goes from 85 hours to 30 hours. I know you're like, well, mine's at 30 minutes. Still, I was excited, right? Because that's a third of the time. And here's what I mean about tiny changes causing drastic changes, right? I reduced it to a third of what it was, and that's what I changed. All of this stayed the same except for that. And just so you can feel sorry for me, that's what I had to make to grow mine. That's a chemically defined medium. Here's what we do for you. Right? I should have worked with E. coli. That's really the take-home message in this. But kind of everything has been done with him. But here's what we feed him. Peptone, beef extract, take a hot dog. We take a hot dog, chop it up, grind it up with a blender, and God knows what's in it. That's what makes them delicious. And then we grow them, right? Whereas I had to meter out all of these different ingredients and pH them. And then when the salt got poured into them, it would cause everything to come out of solution. So I'd have to filter sterilize it and do all this crazy stuff. But at the end of the day, small changes in chemical composition can cause vast changes in the way that these bugs grow. So when we're growing them in the lab, we're trying really hard to optimize things so that they grow quickly, so that we can get results quickly. And thankfully for you guys, most everything we work with, all those 65 organisms on the back, they like hot dogs. They like hot dogs, they like it 37 degrees, they like the pH to be just slightly above neutral, and they'll grow like gangbusters for us, right? One other fun thing about differential media, and then we're done for the day, we also have differential media that are solid, and you're gonna get to use these. This one, by the way, is called mannitol salt agar. It has salt in it. So it will only allow salt-tolerant organisms to grow, like Staph aureus, but it will also allow us to tell whether those organisms can ferment a sugar called mannitol, because if you grow your salt-tolerant, both of these are salt-tolerant, if you are the pathogenic Staph aureus, you will ferment the mannitol and look different. So clinically, once we know you have gram-positive cocci that are catalase producing, we can streak it onto this salt media. And now we know it's not Staph epidermidis, which is normal flora, it's Staph aureus, which could be what's making you sick and causing your boil or whatever nasty thing you've got growing on yourself, right? We have a similar kind of media to tell what's going on in your stool if you have diarrhea. We can grow that stool sample onto a medium that kills gram-positives, because we've got to find some way of wading through all of the little bacteria that are growing in your poo, right? So we kill off all the gram-positives on this plate, and then we add a differential ingredient that allows us to tell lactose fermenters from non-fermenters, because lactose fermenters tend to be normal flora and non-fermenters tend to be pathogens. So if that's growing from your stool, they'll go, hey, you got some Pseudomonas in there. That might be what's giving you diarrhea. And you're gonna get to see both of these media, you're gonna get to use them. You're not gonna bring your stool to class. There are certain microbiology classes that do that. I think that's disgusting, and there's absolutely no pedagogical rationale for you bringing your poop to class. None. Because I worked in a lab for 2 years. No two poops look alike. So there's no reason for you to be like, oh, well, I just had to work with the real stuff. We don't need to work with the real stuff. All right, so that's the end of 6. Here's what you're gonna do. You are going to